Welcome back to our lecture 2, General Botany is our code title, don't forget. And the course code is BOT 120. Therefore, in our last lecture, the first lecture, we talked about the division pteridophyta. Those are the subdivisions of vascular plants. Therefore, at the end of the lecture 1, we talked about the taxonomy of pteridophyta, or the 4 subdivisions of pteridophyta. And one was Coelentophyta, where members belonging to Coelentophyta are said to belong to family Coelentosae. The second subdivision is Lycopodiophyta, where members in this division are said to belong to the family of Lycopodiaceae. The third one is division Equisetophita, where members are said to belong to family Equitaceae, and the last one being Polypodiophyta, where members belong to the family of Polypodidaceae, or it belongs to the families of Traphanes. Now, we are going to start with the first subdivision, and that is subdivision Coelentophyta. I said it is also called subdivision Coelentopsida, or the subdivision Coelopsida. And now you can see an example of Tilopsan plant. They are very small leaves, needle-like leaves indeed. You can see the first diagram there. It's not seen well, but the second one is very seen well. The diagram is said a close-up view of Coelotan plant, meaning that this organism, or the plant organism, belong to subdivision Coelentophyta. You can see it is a spore-bearing plant. At the end, the further end of the leaves, we have the spore-bearing structures. Okay, let's continue. Now, in this subdivision Coelentophyta, this subdivision consists of 2 genera, and the 2 genera are the Coelotan and the Timeris, or the Meris, T silent in this case. And I said these plants belonging to subdivision Coelophyta or Coelentophyta, they belong to the family of Coelentosae. Okay. Now that's something you should not forget. Don't forget that the members of subdivision Coelentophyta belong to the family of Coelentosae. And they have 2 genera, that is Coelotan and Timeris. And now, what is the difference between Coelotan and Meris? Coelotan and Timeris have similarities and they have differences. Now, Coelotan, they lack both roots and leaves. Therefore, Coelotan, instead of having roots, they have structures we call as rhizoids. And according to the formal education in high school, you see rhizoids are used for obtaining of nutrients and mineral salts. Therefore, you have seen that Coelotan lacks both roots and leaves, but are structurally similar to the fossil genus Rhynia. That's Rhynia is a fossil genus similar to Coelotan. Now, what about the Cypteris? Now, the Cypteris has a more complex morphology in that it has aerial roots that are foliar. We have seen that Coelotan have no roots nor leaves. But now the Cypteris have more complex morphology with aerial roots that are foliar. Therefore, what is now the similarity between the Coelotan and Timeris, Mesipteris? Sorry. Now the similarity between Coelotan and Mesipteris is that they have compound sporangia called synangia. That's the main similarity between the Coelotan and Mesipteris. they have compound sporangia called synangia. Now in the case of Slotam, they are three-parted. Now, we have the classification of pteridophytes. Remember, our first subdivision of vascular plants was pteridophytes, and now we have the classification pteridophytes. Now, what do you know about the classification of pteridophytes? Now, pteridophytes, I said both genera, that is Slotam and Metipteris, have aerial branches, and these aerial branches, they develop from stems embedded in a substrate. But now, the other similarity between Metipteris and Slotam is that they lack roots. Remember, I had said earlier that Slotam have aerial shoots, not roots, sorry, but they lack roots. They both Metipteris and Slotam lack roots. But now, the Metipteris are more complex, have more complex morphology in that they contain aerial shoots that are foliar, but they both lack the roots and leaves. Let's continue. Now, how does branching take place in these divisions, the subdivision Coelophyta? Now, the branching occurs dichotomously. In high school, you talked about the dichotomous scheme. Now, branching occurs dichotomously by the division of the apical meristem. Okay, and this is obvious in the aerial branches of Slotam. Say Slotam have branches, but they lack leaves and roots. Therefore, branching, I say it occurs dichotomously by the division of apical meristem within the plant. I said Slotam, as I said earlier, they lack true leaves, but they possess structures which are leaf-like extension of the stem. We call these structures the annation. So, the Slotam lack true leaves, but they have leaf-like extensions of the stem, which are called annation. Now, why do we not consider this leaf-like extension as leaves? Because they lack a vasculature, or they lack the vascular system. Therefore, we don't consider them as true leaves. Are we together? Okay. Let's go to the gametophyte. Now, the gametophyte of the both genera, and when we talk about the genera, we talk about the Slotam and the Metipteris. The Slotam and Metipteris is one. When we talk about the genera, we talk about the two genera, Slotam and Metipteris. And now this Slotam and Metipteris both are non-photosynthetic, meaning they do not carry out photosynthesis. And therefore, that's another similarity. Both are non-photosynthetic, and therefore they live in association we call as fungus. Now, in case of Slotam, the gametophyte of certain twins, they produce vascular tissues. And they said earlier that vascular tissues are the xylem and the phloem. Don't forget, vascular tissues are the xylem and the phloem. And now in the case of Slotam, the gametophyte, we say that there is a gametophyte and a sporophyte. So the gametophyte produce a vascular tissues, that is the xylem and the phloem. Now, that's all about the subdivision Coelophyta. Now we go to the second subdivision, the Lycopodiophyta, which is also known as Lycopodiopsida or the Lycopsida. Now, plants belonging to this Lycopodiophyta as it will belong to the family of Lycopodiaceae. Now, plants have scale like leaves. Now, meaning they have leaves, they have roots, stems, and rhizomes. So, that's the difference between Coelentophyta and Lycopodiophyta. Leaves in Lycopodiophyta plants in Lycopodiophyta have scale like leaves, roots, stems, and rhizomes. But in Coelentophyta, they lack true leaves, they lack roots, and so on. Now, another feature. You can see there is a diagram here showing a Lycopodiophyta. You can see there is a leaf here, and if you see the leaf, it means it has a vascular system. Now, this leaf, it can have a serrated margin. The first one shows entire or smooth margin, but the second megaphyll shows a serrated margin. Now, the macrophyll, it is an unbranched leaf vein. You can see it has only one leaflet at the center, vein, not leaflet, at the center of the leaf. But now in the megaphyll, we have the branched vein. In the first macrophyll, why do we call it the macrophyll? Because it has only one vein passing across the leaf, which has no branched other veins. But the second megaphyll, we call it the megaphyll because it has the main vein passing across the center, but with some branched veins. That's the difference between macrophyll and megaphyll. That's another diagram showing the adventitious roots in Lycopodiophyta. You can see along the stem we have some roots, some root-like structures moving downwards, and those are roots we call the adventitious. So in these macrophylls, you can see it, they have only one vein, as I have said earlier, and are associated with a leaf gap. So they pass across the center of the leaf without other veins. So only one main vein. Now, I say the roots are adventitious. How? You can see the roots are rising from a stem. In the first diagram here, the roots are rising from the stem, moving downwards, where they attach themselves to the soil. And that's what we call adventitious roots. Okay, let's continue. That's about division Lycopodiophyta. Now, what about the branching? Branching in both shoot and root occur by division. I said in Coelentophyta, branching occur dichotomously. But now in Lycopodiophyta, branching of both in shoot and in the root occurs by division of the apical meristem. Let's go. Let me remind you of what we are talking about. Division, Coelentophyta. Branching in division, the third point there, branching occurs dichotomously by division of the apical meristem. But now in Lycopodiophyta, we are saying that branching in both shoot and root occurs by the division of the apical meristem into two parts. So meaning no dichotomous division. It is only division of the apical meristem. Now, the next point about this subdivision Lycopodiophyta is that instead of flowers and seeds, you know the flowers are the reproductive organ in a flowering plant. But instead of having seeds, this division Lycopodiophyta, the plants belonging to the subdivision, they produce by releasing spores. And spores are contained in a structure we call is the sporangia. Sporangia encloses the spores. Now this sporangia, after it ripens, it bursts open, releasing the spores. It's okay. Now these spores, they travel to new places, and after these spores may be dispersed by wind or by water. After dispersion, they are released or they are distributed to other places where they grow into the same species as the original plant, and that's now the dispersion of these spores after they get released by the sporangium. Okay, the next point is that the division as they live in families. The main family, I said it is Lycopodiaceae, family Lycopodiaceae, but we have the other families, the Isostaceae. Isostaceae contain the quillworts family. The division Lycopodiaceae contain the clubmoss family, and the division Selaginellacea, they contain the spike mosses family. Those are the three subfamilies of subdivision Lycopodiophyta. Family Lycopodiaceae, family Selaginellacea, and the family Isostaceae. So, okay, and we have the examples of each family having an example there. In the case of Lycopodiaceae, we have the clubmoss family. In the case of Selaginellacea, we have the spike mosses family, and the last family is Isostaceae, containing the quillwort family. The point number next is the living members are all herbaceous, although isots are secondary growth. What's the meaning of herbaceous? They are herbs. They live as herbs. Therefore, the living members of the subdivision Lycopodiophyta, are herbaceous, except the organism belonging to the family Isostaceae, which shows secondary growth. Herbaceous meaning they live very small in size, but now the family Isostaceae, they grow into a bigger plant species. Therefore, that's why we say the living members are all herbaceous. They live as herbs or as small plants, except the family Isostaceae. Okay, this is an example of a Lycopodiaceae. This is the clubmoss family. This is a typical diagram of a clubmoss. You can see it very well. The green ones are the leaves. We say they have leaves, they have roots, and they have stems. Remember, in Silotophyta we say they have not true leaves, they have not true stems. But now in Lycopodiaceae, they have leaves, they have stems, and they have roots, and also they have the rhizomas. Now that's a typical diagram of a clubmoss or Lycopodiaceae plant. We also have the Selaginellacea, another class of Lycopodiaceae. We have the other class of Lycopodiophyta. We have the Selaginellacea, and that is the spike moss. Typical diagram of the spike moss belonging to the family Selaginellacea. You can see it has leaves together with the branch. So it has a branching to form the leaves and the branches. The other one is here, the third family Isostaceae, composing of the quillworts. Now these are the diagram of a quillwort. All these, the three, that is the Lycopodiaceae, the Selaginellacea, and the Isostaceae, they belong to the subdivision Lycopodiophyta. Those are the three subclasses of Lycopodiophyta, and that is Lycopodiaceae, Selaginellacea, and the Isostaceae. Each one has an example, whereby in Lycopodiaceae, we have the clubmoss family, the Selaginellacea, we have the spike mosses family, and the Isostaceae, we have the quillwort family. Now we have the third subdivision of Pteridophyta, and that is the Equistophyta. And I say the other name of Equistophyta is Equistopsida or the Sphenopsida. Now, Those names have no great importance. You can just get one name, that is the Equisetorophyta or the Equisetolepsida. And now, in this subdivision Equisetorophyta, the division of plants occur with a fossil record going back to a Devonian. Therefore, examples of members belonging to Equisetorophyta, we have the hostels. I said that this Equisetorophyta is a division of plants with a fossil record. Example being the hostels and others. We'll see them later. Now these species, the hostels or the species belonging to Equisetorophyta, I say they belong to a class known as Equisetophaceae. Therefore, these species grow in wet areas with water. Therefore, this means that they only survive in wet areas. They cannot survive in arid or semi-arid areas. Therefore, they have needle-like leaves radiated at regular intervals from a single vertical stem. Therefore, they have needle-like leaves, and these leaves arise from a single stem, not branched stems, only a single stem. Now, examples are here. This is an example. You can see only one stem growing from the soil, and now the leaves being needle-like, they arise at the sides, each side, from the sides of the stem. And therefore, that's where we say the leaves are needle-like, arising from a single vertical stem. Vertical meaning stem moving upwards, no branches, only leaves arising around the stem. And this diagram is an Equisetum telmati. Now, the other example, we have this one. The other one, you can see they are alike. The stem is vertically located, and now no branches, needle-like leaves are rising around the typical stem, which is vertically located. Therefore, meaning there are no branches in subdivision Equisetorophyta. Hope we are together there. Have been understood. Therefore, this subdivision, I repeat, they have needle-like leaves. And these leaves do not arise around branches. They arise around a vertical stem. The stem from the soil, it grows vertically, and now the leaves, the needle-like leaves arise around the vertical stem. This is another one. You can see it, only one stem growing vertically. Same to this one. Another example of Equisetorophyta. You can see the leaves are rising from a single stem. That's the stem growing vertically from the soil. No branches. And now, what about the vascular system? The vascular system itself is curiously, it resembles a vascular plant's ST. So meaning it has a vascular system too, this subdivision Equisetorophyta. They have both primary xylem and secondary xylem. We also have the phloem. Now, in this plant, the cortex itself contains vallecular canals. Now, due to the softened nature of the phloem, these are very rarely seen in fossil substances. I said the development of Equisetorophyta are in form of fossil substances, and therefore you may not see the phloem though it is there. Instead, it contains vallecular canals. Now we have the other point about this Equisetorophyta is the plant does not bear coherent roots system, but now underground rhizome. Rhizomes are structures bearing the They are root-like structures which obtain nutrients from the soil, or they obtain water. Therefore, they do not bear roots. Instead, we call the root-like structures the underground rhizome. These rhizomes are used to obtain water and mineral salts instead of having roots. Now, you can see in this plan we have the adventitious roots we are talking about. You can see them. Now, this rhizome is the structure lying just below the surface of the ground. You can see an horizontal structure. That's what we call the rhizome. This rhizome, they give rise to the rhizoids. And now, these adventitious roots are what we call the rhizoids. The adventitious roots arise from the rhizoid. And therefore, that's why we talk, they do not have true roots, but they have the rhizome. Now, let's continue. I said the members of this class or subdivision Equisetophita are also known as Sphenopsida. And these members, they bear cones. They do not have flowers. They bear cones at the tips of some stems. Not a must to bear the cones, but they may bear fronds at the tip of some stems. Not all stems actually. Therefore, it means they lack flowers. They are cone-bearing structures. The next, now these cones comprise of the sporophores. These sporophores, they are the spore-bearing structures. They bear the spores. Now, remember I say the Equisetophita are arranged into they bear cones. These cones are arranged into sporophores. These sporophores now are the structures which bear the spores. So, okay, let's continue. Now, these spores themselves, they bear characteristics. Distinct, example, they have distinctive, you can distinguish spring-like attachment, which are hygroscopic. Hygroscopic means they absorb water from the atmosphere, but they will not form a solution. Therefore, these spores are said to be distinct from, you can distinguish them. Attachment, they have attachment which are hygroscopic, and this means they change their configuration in the presence of water. They absorb the water and change their shape, though they will not form solution. They will remain to be spores, but they change their shape or configuration in the presence of water. And this helps the spores in their dispersal. After they change their shape, the suitable agent for dispersal disperses them to new areas. Now you can see the meristems. An example of meristem in this division Equisetophita. These are intersecallary meristem. You can see it in the first diagram here. But this intersecallary meristem is only present in a monocotyledonous stem. The cotyledonous stem lack this intercalary meristem. Now we can continue with the last subdivision, and that is subdivision Polypodiophyta. As I had said earlier, this subdivision is also referred to as true ferns. The fern and these allies are put into this subdivision Polypodiophyta, and therefore they consist of a group of non-seed bearing plants. They do not bear seed. And they have a fossil record, the same as the Equisetofighter, and there are over 11,000 living species of Polypodiophyta. Ferns are very many. There are very many species of ferns. That's where we say there are about 11,000 species of this subdivision Polypodiophyta. So there are very many. I say the subdivision is also referred to as true ferns. Now, this Polypodiophyta consists of group of plants with true leaves. They also have true roots and true stems. Remember the others, except the Equisetofighta, were lacking the roots. Except only the Polypodiophyta, the others lack the roots. Instead, they had the rhizomes. But now in Polypodiophyta, we have the roots. We have true leaves, true roots, and true stems. Now, leaves are microphyls, meaning they have branched veins. The middle vein in the leaf, it is branched into many veins, and therefore that's why we say they are microphyls. And in many families, they demonstrate succinate venation. Succinate meaning branching veins. From the middle vein of the root, we have some other branches, sub-veins, smaller veins from the middle leaf. That's why we say they are microphyls. Now, the other point, we have also non-living fern, which demonstrates true second growth. Therefore, this division Polypodiophyta, members of the division, they show secondary growth, consisting of non-living fern. This non-living fern means that they are fern, but they are like dead. They are not living fern, which demonstrate true secondary growth. And they also have the adventitious roots. Roots arising from the main roots. Some adventitious roots arising from the stem to the soil. These members also reproduce by means of spores, which may be either homosporous. Remember we said earlier in the first lecture that homosporous means that they produce only a single type of spores, or they can also be heterosporous, producing two different types of spores. As we said, spores differ in size, and the male spores are microspores. They are very small. Microspores meaning they are small in size, while the female spores are megaspores. Megaspores meaning they are larger in size. So I said the spores differ in size. Male spores are smaller in size, and therefore we call them the microspores. The female spores are larger in size, and we call them the megaspores. Now the homospory. I said homo meaning one. The spores producing only one type of spore. Now the homospory with the production of photosynthetic free living gametophytes is most common in pattern. So the homosporous gametophyte is more common in Polypodiophyta, producing photosynthetic free living gametophytes, so they can carry out photosynthesis. And now the structure bearing the spores, we call it the sporangium. This sporangium, it encloses the spores. When it ripens, it bursts open, releasing the spores. And therefore, the spores are contained in a structure we call as the sporangium in the subdivision Polypodiophyta. Remember, these subdivisions are subdivisions of division Pteridophyta. Members of division Pteridophyta, we said earlier, they include hostels, mosses, liverwort, and many others. And this is an example of Polypodiophyta. You can see it has true stems, true branches, also true leaves. And now underground, you cannot see the leaves, the roots, but they are also present. Now, let's go to the other topic, the fan structure. Now, fans, like the sporophytes of seed plants, fans consist of stems, rhizomes, stolon, trunk, and many others. These now are fan structure. I say remember we have a leaf known as frond. Now, on top of the leaves, we have some seed-like structures known as sori. Sori, I said, is in plural. Singular is sorus. You can see the sori on top of the plant. You can see the structures here, the sori, the seed-like structures on top of these leaflets. I say the leaflets, we call them pinna, are known as the sori. This sori, they are the one enclosing the spores. Now, they enclose the spores. I say the leaflets are known as pinna. When you talk about rhizome, this is now the rhizome here, the one containing the roots. The vertical thick stem here, it's not actually a stem, it's what we call the rhizome. And therefore, this rhizome is present in the fans. We say fans are like other sporophytes. Sporophytes meaning spore-bearing structures. They have rhizomes, they have the frond, they have the sori, they have the pinna. Meaning they have stems, they have the rhizome, stolon. Stolon is seen below the surface of the soil. They have the leaves. You have seen leaves here called the fronds with the leaflets. We call them pinna. Now, I've already said that the ferns have leaves known as frond. Now these leaves, they have the leaflets. We call the pinna. We say that the leaves are pinnate. They contain some leaflets, smaller ones. Now, these leaflets, if you observe them using a microscope, or if you find a typical fern around the rivers, you will find that they have a succinate venation, meaning that the veins are rising, that they have branching veins arising from the middle vein there. We have the branches of veins arising from the main vein. Now we have other veins around the middle vein from the leaf. And therefore this is what we call succinate venation. I can show you an example. If we get a diagram with that kind of venation, we said that at the middle of the leaf, we have the main vein. From the main vein, we have the branching veins there, going horizontally. That's what we call succinate venation. These are young and rolling front. You can see the fronts here. These neck-like structures are young rolling front, not plant, sorry. Now, leaves are divided into two types, and that is trophophil, and the other one is sporophyll. Trophophil and sporophyll. Now, trophophil, I say leaves in ferns are divided into two types, trophophil and sporophyll. Trophophil is a leaf that does not produce spores. Trophophil are leaves that do not produce spores, but instead they produce sugars by photosynthesis. So trophophil do not produce spores, but they carry out photosynthesis, producing sugars. Now sporophyll, they produce spores, and these leaves are analogous to the scales. They produce spores, they do not carry out photosynthesis. Now, we say there are two types of fern leaves, trophophil and sporophyll. Trophophil carry out photosynthesis. This is an example of trophophil. You can see it has green coloring matter, and that's chlorophyll. Therefore, it carries out photosynthesis. Now, sporophyll, is here. Though it is green in color, you can see on top of the leaf, we have the sori. Remember I said sori is in a plural, sorus in singular. Now this sori, I said are spore-bearing structures. After they ripen, they burst open to release the spores. Therefore, the first diagram here shows a trophophile fan front. The second one shows a sporophile fan front. The first one you can see it is airy-like. That's why we call it airy-like trophophile fan front. You can see some structures like hairs. That's why we say airy-like trophophile plant. Trophophile meaning they don't produce spores. Now in the second diagram we have the sporophile fan front, meaning that they produce by means of spores. Now, remember we are talking about the structure of fans. We have stems, leaves of two types, sporophyll and trophophile, roots. Now roots are always fibrous roots. Remember, fibrous roots are present in monocotyledonous plants. So many fans are examples of monocotyledonous plants. Therefore, they contain fibrous roots. And these roots are very similar to the roots of seed plants. Therefore, what you should know is that the roots in fans are fibrous roots. Now, what about the gametophytes in fans? Now, the gametophytes of fans are very different from those of seed plants because it consists of something we call as a prothallas. Now, this is the shape of a prothallas. This is the shape of a prothallas. You can see it. If you observe it well, at the top you can see the archegonia. At the bottom is where the antheridia, we talked of antheridia and archegonia are located. Now this prothallas, I said, it is present in a fan. Remember we are talking about fan structure. It has a gametophyte, it has stems, it has roots, and it has leaves. Now prothallas is now the gametophyte. Gametophyte, we said it produces a structure we call as the prothallas, and this prothallas is the one containing the gametes. Gametophyte produce prothallas. Prothallas contain the gametes, and the male gametes we say they are known as the male structures are known as the antheridia, and the female structures are known as the archegonia. The antheridia are the one which releases the antherozoids, while the archegonia they releases the egg cell. Now, this gametophyte, apart from the prothallas, it also contains the rhizoids. Rhizoids, let me show you the prothallas together with the rhizoids. Now this is the prothallas. Now you can see very well. It has the male structures and the female structures. The male structures is the antheridia. At the bottom here you can see the antheridia. They release the male gametes known as the antherozoids. Above the antheridia we have the archegonia producing the egg cell. Now you can see the root-like structures just below the prothallas. Root-like structures, we call them the rhizoids. And that's why we say the fan contain gametophyte, and this gametophyte contain the rhizoids and the prothallas. Let's continue. Now the prothallas, it is green in color, as you can see. Here it is. Prothallas is green in color. And if you see green color in plants, it means that it can carry out photosynthesis. Therefore, photosynthesis takes place due to its photosynthetic structure of the prothallas. And the prothallas, look at the shape of the prothallas. This can show you the shape very well. It looks like a kidney or the heart. Therefore, it is heart-like prothallas. is heart-like or kidney-shaped. It is 3 to 10 millimeters long, and the broadness is 2 to 8 millimeters. And therefore, as I said earlier, the prothallus produce gametes. And these gametes are produced by the antheridia. I said the antheridia are small spherical structures that produce not a sperm for sure, but it is the antherozoids. Antherozoids are the same as sperms in human beings. Frangulate sperms are what we call the frangulate sperms, the antherozoids. Now the archegonia, they are flask-shaped. I said the antheridia are spherical structures, but now the archegonia, are flask-shaped. And now they release zygote egg cell. Antheridia release the antherozoids, while the archegonia releases the egg cell. And you know, if the egg cell and the antherozoids fuse, they form the zygote. Now let's continue. These are the results we are called. The results, I said earlier, I can show you they are root-like structures in the prothallus. Let's go back to it. These are like roots. They are known as roots. Now these roots, we call them the rhizoids. They are not true roots, but they are known as rhizoids. They are used to obtain nutrients from the soil. Now let's continue. So they are root-like structures, and I say they are not true roots, rhizoids. Therefore, they consist of greatly elongated cells, and they absorb water and mineral salts, I had said. The other use, I said the first use of rhizoids is that they absorb water and mineral salts. The other use of the rhizoids is they are used for anchorage. They attach the prothallus firm into the soil, and therefore that's what we call the two uses of rhizoids. They absorb water and mineral salts, and also anchorage, attaching the prothallus firm into the soil. Now these are very typical plant of a fern. You can see the rhizome. When we are talking about the rhizome, is the black. You can see very black. The rhizome, it provides a surface for the growth of the stem, and under the rhizome is where we have the roots, but we say they are not roots, we call them the rhizoids. We have also the leaflets, we call them the pinna. You see they have leaflets known as the pinna. Now this is how the stem looks like of a fern. This is the stem of a fern. It has the middle stem here with the leaves arising from the stem, and this branch, it has its leaflets where we call them the pinna. Now that's the end of lecture 2. Let's meet next time for lecture 3. Don't forget, my name is Dr. John Musau. I wish you all the best. Thank you so much for cooperation. We'll meet next time.